Reproductive cloning starts with a cytoplasmic donor, shown in the upper left. In the case of Dolly, the first cloned mammal, the cytoplasmic donor is a Scottish blackface sheep. The cytoplasmic donor donates a cell from his or her body, and it can be any type of cell because every cell in the organism has the DNA needed to create an entire new organism. The DNA is removed from the cell in a process called enucleation. In addition to a cytoplasmic donor, a nuclear donor is also needed, shown in the upper right. A nuclear donor is a female and donates an egg. This egg is also called an oocyte. The nucleus from the oocyte is removed and the nucleus from the donor is inserted. A new cell is now produced. The new cell is now given a small pulse of electricity to bind the nucleus to the enucleated oocyte's cytoplasm. In addition to binding the nucleus to the cytoplasm, this also helps start the dividing of the cell. The new cell is now placed in a female sheep's reproductive tract to grow. After 148 days, the normal length of time, Dolly was born. Dolly has the same DNA as the original donor. Therapeutic cloning is performed not to produce another organism, but to harvest embryonic stem cells for use in medical treatment. Embryonic stem cells are those cells found inside developing embryos. They can be used to produce a number of different cells, including connective tissue, nervous cells, or even blood cells. Just like reproductive cloning, a cell is removed from the patient requiring medical treatment. This is the cytoplasmic donor. The nucleus of the cell is removed and is inserted into an empty donor egg, the nuclear donor. Division is encouraged through the use of special chemicals or an electric current. Now this produces a blastocyst containing stem cells. Up to this point, therapeutic cloning is the same as reproductive cloning. However, after this point, it is entirely different. Rather than inserting this new cell into a reproductive tract to grow, it is placed into a petri dish and stem cells are produced. Also, rather than producing a completely new organism, these stem cells can be coerced into becoming blood cells, cells of the nervous system, connective tissue, or really any body part. The question of whether therapeutic cloning is infanticide brings up the issue of, is an embryo human? Because people say that if an embryo is not human, then it is morally justable to harvest stem cells. The qualifications for a human are that one must think and reason independently, exist independently, and have a digestive system for nourishment. An embryo can do none of these, but that does not mean that all humans can do them as well. Some children are actually born with a portion of their brain missing and cannot totally think independently. Others are born with hydrocephalus, a condition that severely compresses the brain due to a buildup of liquid. This restricts the thinking power of the child. No one would ever claim that they are not human. The fact that an embryo cannot exist independently does not just apply to embryos. If a newborn child was not nurtured by its parents, it would surely not live. However, no one would ever consider a newborn baby not human. This also applies to the terminally ill and disabled. Some people are born without the necessary organs, or because of the disease need to have them removed. The clients who need a new organ from therapeutic cloning may even need it because they cannot properly digest food and get nourishment. Nobody would call these people subhuman or not human. So saying that an embryo is not human is a very false argument.